So I'm sure all of you have heard about variants, Omicron, others, sequencing, mutations. So basically all of you are genomicists. You know what a variant is and how all those things vary. In this figure here, this is the same person, right? But then without enough resolution, you'll say they are the same person, right? But if we sort of have more light, they're different individuals, right? So this is the power of genomics. It sort of sheds light into the invisible. If things are dark, just get genomics. You see everything in clarity. So how do we use genomics to understand AMR across scales? So I'm going to walk you through Biology 101 of AMR, and then give you examples of how we apply genomics in hospitals, in communities, and in understanding transmission dynamics of AMR. So the first thing is that you cannot eradicate AMR. Bacteria have been in this world longer than us. It's an arms race. They're trying to live. They have brothers, they have sons, and they have aunts. They're trying to live. We are trying to fight them through antibiotics, and we can't win the war. We are losing the war. We can't eradicate AMR. We can only manage it. In this recent global review, they found that just under 2 million mortalities per annum is due to AMR, direct mortalities. Just to give you a flavor of what that means, look at HIV, under 1 million. And this number is meant to grow all the way to 50 million in the next few years. Therefore, all of us should be worried about this problem. Not just about dying, it's about economics. Think of livestock farmers unable to treat the animals. Think of trade implications when it comes to AMR. It's going to have a huge direct and indirect cost to communities. And then, of course, as always, it's going to affect poor areas just simply because of lack of very strong systems. And again, I'm now 30 plus years old. And the last time there was a new antibiotic was when I wasn't born. So you can imagine, there are no new antibiotics. Therefore, we have to manage what we have. Look at this figure from this particular paper there. They show that the highest level of mortality was in Sub-Saharan Africa, Southeast Asia, and Central and Eastern Asia. Again, showing you where the problem is. Closer home, this was a few days after my son was born. We lost 6 babies at our national hospital because of drug-resistant Klebsiella in Nairobi. Just showing you how of a problem this is. So again, Biology 101 of what AMR comes into being, 2 ways. One, you go to a pharmacy, you get an antibiotic. This is the bacteria in your gut. One of them undergoes Darwinian selection and then becomes resistant. And then that passes through generational mutation and growth. So the main driver when it comes to AMR is antibiotic use, whether you call it use, misuse, or underuse or overuse, whichever way you look at it. It's about use of antibiotics in human and veterinary systems. How are the patterns looking like? In this paper by our colleagues in the US, they found that the levels of antibiotic usage are increasing, actually exponentially, if you look at it in this category of low and middle income countries. It's plateauing a bit more in the high income countries, but this is very worrisome because we heard from Marcy that urbanization is leading to all those things. And therefore, we are going to see a lot more of resistance due to this. And then the second pathway is transmission. We know that we don't live alone. We live with animals, right? We live in the same ecosystem with animals and other things. The second thing is transmission. We use antibiotics in humans and in chickens and cows, right? And we live with them, right? You may not live with a cow, but you eat chicken, or you share the same environment. So there's transmission of the whole bacteria. So E. coli moving between chickens and humans, or extra chromosomal elements called plasmids, which don't require the whole bacteria. So plasmids there are phages, transposons, and others, which are moving in the community within humans, within animals, and then in hospitals. And then the second thing is now these plasmids, and this is the most worrisome one because it doesn't necessarily require the whole bacteria. Therefore, there's very minimal ecological fitness when it comes to moving. So you can see before and then after there's acquisition of that particular element. And then the other problem is access versus excess. We should be very careful to say, let's not reduce antibiotics because the highest cause of mortality in children in Africa is pneumonia. So availability of the simplest molecule called penicillin is the highest cause of mortality in children in Africa. Therefore, as we try to reduce excess, we should also think about excess. Hence why AMR is a huge, huge problem. So I'll give you examples of how to apply genomics in understanding things. And the first thing is trying to understand interventions and dynamics at local scale in hospitals, for instance. So genomics has been very critical in identifying and reconstructing hospital outbreaks. In this study by colleagues in the Gambia, they found there was a huge problem with mortality in children in the hospital. So what they did is that they took the isolates, unknown what they are, they sequenced them, and they found two culprits. One is called Bacalderia, not very common bacteria, Cepacia, and then a common culprit, Klebsiella, which caused the deaths in our hospital here a few days ago, Klebsiella. So by doing genomics, they were able to identify actually this bacteria has been in the ecosystem for the last 20 years, whereas Klebsiella is just a new entrant into the party, and therefore showing how genomics is very critical in understanding some of these local problems. Another example in Pakistan, cockroaches. Not very good friends of ours, aren't they? They found that in a surgical unit in Pakistan, the main reservoir of AMR determinants was actually cockroaches, right? You didn't think of cockroaches as a harm in a hospital. Well, you should, but ideally you shouldn't. You wouldn't probably. And then houseflies. In this example here, we see humans and a patient and a cockroach separated by just 5 nucleotides, therefore saying that the E. coli in humans and the E. coli in chickens, they're basically identical and they're carrying AMR genes, just highlighting how we need to use genomics to understand some of these contexts. Next, again, colleagues in Oxfordshire, they found that it's not just about the bacteria, right? It's about the plasmids. So all of us, when you were learning microbiology in school, we learn about bacteria and why we should prevent bacteria. But really, you think about plasmids. So these plasmids they're like beings. They don't require the bacteria. They're like complete beings. And they found that plasmids were responsible for transmitting enterobacteriaceae in the hospital as opposed to the bacteria. And not just within one bacteria, but they can cross from different bacteria, from E. coli, Salmonella, and other bacteria. Just highlighting the need to use genomics to understand that. Without genomics, you can't be able to know that. And then lastly is to inform diagnostics and patient therapy. There's a big push, at least in the Northern Hemisphere, to sort of move away from basic microbiology, whereby you come in the hospital, you have sepsis, and they just mash your blood into some machine, and it gives you a phenotype within minutes. And that's just the potential of genomics in that. Right. The next thing is these lovely global maps. We love maps, all of us, don't we? At least in epidemiology. So how do these things move? And how can we understand movement of Klebsiella in this part of the world, here and here? The only way is to use genomics. In this paper by Baker et al., he was able to show that by using genomics, you can understand the clonal expansion. Clonal means movement of the whole bacteria. For example, in Klebsiella ST258, it first occurred in the US, somewhere in the East Coast, and then you can see how it spread over time to cover almost the entire world by genomics. Here, you can see an example of that's Salmonella typhi from India, and now it's actually encompassing the entire African continent and others. So genomics gives you the power to sort of map those dynamics. And then even further is now being able to go back and ask yourself, when did this happen, right? So to do that, again, genomics is able to reconstruct phylogeographical patterns of pathogens. In this example here by a group in the UK, what they found is that by using genomics, they're able to identify that Salmonella typhi serotype 558 first occurred in India in 58 years ago. But then over time, it's moved across all these multiple introductions. I mean, the weight means how strong the dispersal is. And you can see even actually with dates when this occurred. And if you overlay this with global travel, globalization, and all these other things we try to do in ecology, you'll see this pattern actually fits quite nicely when it comes to movement of things. And this is the power of genomics in AMR. So now what I do, I don't work in the hospital. So the next thing is now interfaces. And interfaces are those lovely maps you saw from Dino's talk yesterday and other presentations. So an interface is a point where independent systems or diverse groups of systems interact, right? Think of it as a rural-urban interface, right? Think of it as a livestock-human interface. human food interface. When you go for lunch, that's an interface. Chicken and human, it's an interface, right? Pets, the lovely mouse we saw from Anne's presentation, that's an interface, right? And all these other interfaces. It could be socioeconomic interface. So, and these interfaces, these are points where there's disturbance, right? You go to a forest, interaction, right? Genetic exchange and pathogen transmission. An example of this photo in Western Kenya, you can see there's a young boy living with, basically they live with the cows in the same compound. That's a crucible for disease emergence. How can we understand how things are moving between populations? The only way I can think of is genomics. I'll show you why. Again, in this photo in Cambodia by a colleague of mine, you can see it's a wet market in Cambodia. You can see the possibilities of things moving between and across populations. And the power of genomics is what enables you to do that. So again, just a quick flavor of, again, how people have used genomics to define interfaces. If for you work in the AMR world, you must have heard of a new gene discovered in China, conferring AMR to colistin, apparently the last antibiotic of the human race. And in 2019, they found a gene which confers resistance in bacteria against colistin. Colistin is an antibiotic when you're just about to leave this world. A doctor gives it to you, and then you come back, right? And they found a gene which can move between humans, livestock, bacteria, and anything and everything in 2019 through genomics. People went back and looked in the freezers and also surveillance, and they found that gene was across the entire world. South Africa there. I mean, Africa doesn't mean it's not there. We just haven't looked enough. It's all over the world, and that was in 2019. And then genomics enables you to understand the nitty-gritties of variation when it comes to interfaces, the fine-scale epidemiological connections, and the transmission dynamics, whether it's from chickens to cows and the converse. And then lastly, environmental monitoring. In this paper by a group in Denmark, using sewage, they're able to show that we have highest level of resistance or AMR in the African subcontinent. So I'll give you a quick overview of some work done in Nairobi. And Nairobi is very complex. I'm sure you've seen that, right? You see poverty against rich in the same, just separated by a road. You see very complex value chains, movement of milk and other things. You see people keeping chickens and cows. If you haven't seen a cow, just drive around, you see one. This is a city which typifies any other developing country context, right? It's just a sea and a soup of many things happening at the same time. How can we track movement of things? Assume you place a disease in Nairobi, and actually COVID is a good example. How would things move across that very complex system? So, and then we use E. coli to understand how things are moving. So if for us, E. coli is a proxy of movement, right? Because this bacteria is present in everything. If I swab your hands, anything, the floor, food, E. coli is present. So we used E. coli to understand some of those complex dynamics. And our hypothesis was very simple. Everything is everywhere. E. coli is everywhere. And then there's some, for those who are in ecology, you've heard about everything is everywhere, the environment selects, right? It's an hypothesis back in the day. So we use that hypothesis to understand how are things moving. And we hypothesize that distribution of bacteria strains and their genetic pools are limited to particular defined ecological niches, household or host, or they display an everything is everywhere distribution, right? So this is the household, just follow with me. We set up an experiment in Nairobi. This is the map of Nairobi, this is National Park. We are somewhere here. So the green it is, the poshier you are. So we are here. Nairobi is not just all of it green. Nairobi is very different. So green means poshier, red means a bit less affluent areas. So we sampled across the entire city. We sampled cows, chickens, anything and everything you can think of, apart from insects. Right? You can see it's a lovely pig there, some beautiful looking chickens. A colleague of mine collecting environmental samples. So we got all those samples. We did the normal microbiology, genetics, and then fused with phylogenetics. Then those are some of the examples of populations we found in their numbers, humans, livestock, wildlife. We sampled, I think, more than 20 species of different wildlife, right from bats to rodents and carnivores, and just one lovely monkey, I think. Right, so what did we find? So I won't give you the super hard details, just give you a flavor of what we found. What we found was very simple. Don't worry about the phylogeny, just to show you that we did some work. The conclusion is all of Nairobi, all of the world is in Nairobi, and all of Nairobi is in the world. What do I mean? That this is all the possible kinds of E. coli we have in the world. They are all present in Nairobi. Think of COVID, all the possible variants of COVID, they are all in Nairobi, right? And then all the possible COVID variants in Nairobi are present in the world, right? So showing you that the diversity of this species is very important to understand, because it just shows you how, if you are to address a public health problem, how you need to think about it. There's no clustering by host. It didn't matter whether you're a cow or a chicken, you add that particular E. coli. Whether you are rich or poor, E. coli is democratic. It cuts across the wealth continuum. The second finding was, are we sharing things with our compatriots? Are we sharing things with livestock and wildlife? And we used two measures of distance of sharing. One is a CGM, well, I won't give you the details. So that is a nucleotide-based distance. And then we used something for the AMR genes to understand what is being shared. So simply what we found is that sharing of bacteria was shaped by the household and the host, which is very interesting that sharing of things occurs within the household confines. And again, think of COVID. Or was COVID happening within your household, right? But then there are a few instances of inter-household transmission, right? The same case for hosts. We found that E. coli was more likely to be found within chickens than chickens and livestock. But there were cases of cross transmission. And then what about the determinants of resistance? What about the AMR genes? The AMR genes, unfortunately, don't follow those patterns. They cut across. They could move between chickens and cows and anything else. So the distribution of those genes was not limited within the household confines. It was cross-cutting between inter-houses and also across host populations. Therefore, highlighting that when it comes to AMR, it's not about the bacteria, it's about the genes, because they can move up and about without the bacteria. How about keeping livestock, right? Whether you're keeping, so these are cases of humans and livestock sharing the same bacteria. We only found 10 out of 1,000 plus, where you have similar bacteria with a cow or a chicken. So keeping livestock was a driver of sharing a bacteria with your livestock as a human, right? But not always. There were some people here not keeping livestock, and they were sharing bacteria with the livestock from a different place. But there's a problem here. It's all the males. So the men in this hall should be very worried that if an E. coli has to come from a chicken, it probably land in you. So the risk factors here were just very unclear, that mostly males, and also keeping livestock was a risk factor for acquiring bacteria. But we found that manure was responsible for increasing the likelihood of human AMR. So not the livestock itself, but the manure, right? And therefore highlighting the environmental aspect of this. So in conclusion, the dynamism of genomics, it sheds light on knowns. There are things we know, right? Known and knowns, there are things we know, we don't know. And there are things we don't know. And the only way to identify those things is genomics. That is the torch we need to guide our path when it comes to the next 20 years of AMR. But then scaling up, that was a project, for example. These are small projects. How do we scale up this tool across systems? In hospitals in Kenya, in the Gambia, in Congo, and other places. There are things we need to think about. One is, what is the business case for genomics? Why should a government with many other problems like malaria, HIV, TB, tuberculosis, and others invest in malaria? We have to provide a business case for that. And COVID is a good example. Then investment studies as well, ethical issues, data sharing, transparency, right? You can't be sending samples just to a lab in the US without a proper understanding, right? That's very important. Issues around ethical issues. We saw in COVID, naming and shaming of countries using genomics to identify variants, the South African variant, right? So genomics is very good, but the issues we need to think about. Integrating genomics surveillance across systems. Let's not think about genomics just in hospitals, but genomics in hospitals, in veterinary health, and also environmental monitoring. By doing so, you get the benefits. And then lastly, some of the traditional barriers. Infrastructural barriers, like labs. Our labs are not very good. Personnel, training, bioinformatics, all the AI you had in the first day, bring it to AMR, please.
And then supply chain, bring in a sample in our lab at Tilbury from Oxford in the from fish in the UK. It takes 3 months, right? We have samples, they're expiring because we're lacking another sample. Just showing, another consumable, highlighting the need for very clear supply chains. And then lastly, the issue of governance. So thank you so much to the conveners, and yeah, thanks.